Okay. So this chapter was actually, I thought, easier to read than the last one, and fairly straightforward, understandable. So I'll try and review some of the details today and things that I think you should probably put in a binder to memorize for your board. So of course today we're talking about T-cell mediated immunity, and then next week, my last week, I'm teaching is going to be on humoral immunity. Okay. So today's whole chapter picks up where last week left off, right? So we stopped at the naive T cells. And today we're going to talk about when they first get primed, or when the naive but mature T cells actually see antigen for the first time. And then we will talk about the interaction between the effector T cell and its target cell. Really just a word about memory cells. And that's it. All of this together we call cell-mediated immunity. So even though B cells, of course, are a cell, we usually refer to that as humoral immunity because the antibody, that's the functioning part of the immune system in that case, and the T cells have, in a way, directly act upon its target cells. So we call this cell-mediated immunity. T-cell mediated immunity. So as we kind of touched upon at the end last time, there's several different types of T cells. CD8 T cells, the cytotoxic T cells that are directly cytotoxic to other cells. And then there are lots of T helper cell subtypes, such as Th1, Th2, and Th17, all of which we'll talk about a little bit. There's a T cell called a follicular helper T cell. That was touched upon in this chapter a bit, and then, of course, regulatory T cells that are T cells that suppress the immune response rather than instigate it. Okay, so when a T cell leaves the thymus, where we stopped last week, the T cell gets back into circulation. And where it needs to get are the lymph nodes or the peripheral lymph tissue or the secondary lymphoid organs. So the point of the secondary lymphoid organs is to bring T cells and the antigen-presenting cells into close quarters with each other. So if all of these things were just floating around in the blood vessels, they would never be able to see each other. So I call it the dating bar of the immune system. This is where everything needs to go to meet up. It puts them in close proximity to each other. So how is this going to happen? Well, the T cell is going to be traveling through the blood vessels, and when it comes to a lymph node, which is going to be the example we use most of the time here, it escapes out something called the high endothelial venules, or HAVs. And then it will end up in this T cell zone of the lymph nodes. And the lymph nodes, remember, the architecture is such that there is a B cell zone and a T cell zone. So in this T cell zone here, it can look around and sample and look and see if any dendritic cells are holding an antigen it is specific for. If it's not, then it just leaves through the lymph. But if it is, if it's activated, if it sees something it's specific for, then this is when everyone's going to take place. It's going to proliferate. It's going to decide its effector function. And then it will still leave in the lymph, but instead of recirculating from node to node to continue sampling, it's going to go leave the lymph node system altogether and go to the site of infection to do its effector function. Okay, so exiting at these high endothelial venules has been a focus of a lot of research. So just like when something's trying to slip out of a vessel wall, this happens by a process of slowing, stopping, and then slipping through two cells. So the rolling like we've talked about before, is mediated by selectins. So selectins slow the vessel along the wall. In this case, there are L-selectins that are actually on the lymphocyte. Now it's opposite at the site of infection, where E and P selectins are actually on the endothelium. So don't get those confused in your head, as I did when I was taking my board. L-selectins are on the lymphocyte. Either way, the selectins slow. And then because of the chemokines in the area, so in this case it's going to be chemokine CCL21, which binds to CCR7. So CCR7, remember, is the address to get to the T cell zone of the lymph node. And that's going to cause upregulation of integrins and integrin ligands, which is going to cause a stable adhesion. So it's going to roll and stop because of the integrins. In this case, LFA1 is one that was covered a lot in this chapter. And then it's going to flip between two cells in a process called diapedesis and continue going to where it needs to go to within the T cell zone. So this actually, I think, was quite a bit better covered in Abbas in Janeway than it was in Abbas. It really, I think, handles the integrins and selectins better. So they did a great job in pointing out which ones would probably be good ones to commit to memory, and I think this table sums it up very nicely. Very important, of course, is LFA1, the integrin LFA1, which has CD18 on it, right? The adhesion molecule causes adhering. But of course, if it's associated with an immune deficiency, you know you need to know it. So LFA1, which binds to ICAM. And then LFA3 or CD58, which is called more in this chapter, binds to CD2. And then there's another integrin called VLA4 that occurs on activated endothelium. So the VL is very late. So after the immune response has gotten going, in the very late stage, this is one that's upregulated. So I think that's an easy way to remember. VLA4 is on the endothelium. It's not in the early stage on the lymph node, in the early stage defense. VLA4 binds to VCAM1. In the case of ICAM1, VCAM1, I don't necessarily think you need to know the CD markers on these. I would certainly know, however, again, what integrins have CD18 on them. Okay. So once again, some integrins that are important to know. LFA1 binds to ICAM1. So LFA1 is made of both an alpha and beta subunit. It's found in the high endothelial venule. And then on the activated endothelium, like I mentioned, there's, of course, a beta and alpha subunit, VLA4, and VCAM1. I'm pointing out this one because this is actually the only set of subunits that I see named specifically. Alpha 4 beta 7 binds to MADCAM1 on the mucosal endothelium. I bet the MA is mucosa associated, although I don't know that for sure. Alpha 4 beta 7 MADCAM1 at the mucosa. So this is an interaction, and I would know this interaction, and I would know it's alpha 4 beta 7. I have rarely heard LFA1 or VLA1 called by its CD marker or by its subunits. Okay, so let's take a look at what's happening. So you have your lymphocyte. It's at the high endothelial venule. It's deciding to get out at this lymph node. So it's coming along, and then the L-selectin is on the lymphocyte, is going to interact with its selectin ligand. In this case, it's glycam1, and it's going to allow for slowing. So these are going to be several quick on-offs. So it's just going to slow down, and then you're going to be able to have a signal sent from the cytokine in the area, right, in this case in the lymph node. CCR7, you want it to come out, CCL21. And then the integrin, an integrin ligand, is going to be upregulated. You're going to have a tight adhesion, and it's going to stop because of that tight adhesion. Once that happens, the lymphocyte can actually migrate out. This whole process we've talked about again and again, but very important to know. This time through, since I think we probably have the basic concept down, I'd probably spend more time concentrating on whether each component of these is on the cell or on the vessel and what they're called. I think those things are very easy to get mixed up in your head, but at this point, very important. So earlier I mentioned if a lymphocyte, if a T lymphocyte samples around and is not activated, it doesn't see antigen specific for it, it goes back out, it exits the lymph node, goes back out through the lymph, and then eventually back into circulation. And one of the things that mediates this process is called CYP, or CYP1, sphingosine 1 phosphate. We call it a CYP1 gradient. So the naive cells, express the CYP1 receptor. So if it hasn't been activated, it's going to be drawn away from the lymph back into circulation. So the CYP signal is coming from outside the lymph node, and the lymph node is going to sniff towards it. But then, as you would expect, activated T cells, they lose the CYP receptor because they want to be retained inside the lymph node where it can have time to fully differentiate, fully activate, activate B cells, and basically do everything it needs to do in the lymph node before it's completely ready to get to the site of infection. We know this takes some time because if you get an infection, the whole process, your whole healing process, especially the first time around before you have memory, it takes some time. It takes several days, right? So it takes some time for the T cell to find the antigen and then to get fully activated and to work and then get to the site of infection. So it takes time. So you want that T cell to stick around for a little bit. So it loses its CYP1 receptor so it doesn't leave until it's absolutely ready. Once it's completely activated, ready to go, then it will get it again so it can roll on out. Okay. So a T cell, once it's into the lymph node, so there's 3 different cells that are specialized to present to it: dendritic cells, macrophages, and B cells. Now remember, any cell can present. Many cells, almost all cells, except ones that don't have a nucleus, like red blood cells, will have MHC class 1, so they can present to T cells. However, there's only 3 cells that are specialized to present to T cells and have MHC type 2 as well as MHC type 1. These are dendritic cells, macrophages, and B cells.
I've been seeing some of your research looking at eosinophils being able to antigen-present cells with C2 possibility.
Yeah, very possible. I would buy it.
Interesting fact.
Yeah. I wonder if mast cells too. I think of them as fairly equivalent to macrophages. Anyway, neither here nor there. So what the cell, though, that really gets the process going, that changes the naive to effector cell, is the dendritic cell. So this is the antigen-presenting cell that's important in this case. For the most part, macrophages and B cells just sustain effector cells for as long as the immunologic battle needs to occur. Today, the vast majority of the focus is on dendritic cells. So I think we talked about this earlier, but there are 2 types of dendritic cells: conventional dendritic cells and plasmacytoid dendritic cells. Actually, I think there's 4 subtypes of dendritic cells. The difference between conventional and plasmacytoid is its function. So plasmacytoid specializes in secreting interferon, and that's how it does its job. Conventional dendritic cells specialize in presenting to T cells. can tell them apart by their various cell surface markers, right? So you would anticipate conventional dendritic cells to have lots of MHC class 1 and class 2, as well as co-stimulatory adhesion molecules to allow the entire response to happen. So the dendritic cell is kind of the sentinel. So it's the one that's going to be sitting at sites where infection may occur. So in the case of the skin, right, we call them Langerhans cells that have the Burbeck granules inside. And when they see an infection, they're going to engulf it in order to process it along that MHC2 pathway, or MHC1 pathway, depending. Right, and it can do this in many different ways. So one way is receptor-mediated phagocytosis. So we know there's lots of different receptors on the outside of dendritic cells, like toll-like receptors, that can directly recognize a pathogen. If it binds to it, it can engulf it. Of course, also a process of macropinocytosis, when it basically just engulfs a large amount of cytosolic fluid with the pathogen right inside. Of course, the dendritic cell itself can be virally infected and present on an MHC class 1. But then dendritic cells too, and I would know this is another, just a little bit of a tricky detail. Dendritic cells can do something called cross-presentation. And this is interesting because the virus gets engulfed, whether by macropinocytosis or by receptor-mediated phagocytosis. And when things are typically taken up in this way, you anticipate them to get loaded onto MHC class 2, right? So that's how we learn those pathways. It comes from these vesicles onto MHC class 2. It doesn't go through that CAT system. But in this one circumstance, even though it's engulfed and you'd anticipate it to go through the MHC class 2 loading, it actually gets loaded onto MHC class 1. And this makes sense because what you want to activate is a CD8 cell, right, which are very good at virus fighting. So you want this to happen. It's a little bit counterintuitive because it's engulfed, but it's viral. It still gets loaded onto an MHC class 1 and presented. And then there's some suggestion that dendritic cells can actually directly transfer between dendritic cells. But I don't think we actually know too much about that. I don't anyway. So after this happens, after, say, a Langerhans cell, skin, or other dendritic cell, engulfs, ingests, it's going to jump out of the area it was, the skin, and head back through the lymph to lymph nodes. So this is going to occur based on the same address. So dendritic cells want to go to T cell zone, so they're going to have CCR7 receptor. And it's going to get them to the same place that T cells are where they can meet. And as this is happening, the dendritic cell is packaging onto MHC class 1, MHC class 2. It's presenting it. It's upregulating its co-stimulators, and it's going to be ready to present to that T cell. Right, so here it is once again. You have your pathogen, which is recognized and engulfed by the dendritic cell. Using its address, it's going to go to the T cell zone of the lymph node. And then when it finds a T cell, it's going to present to that T cell. And if that T cell does not recognize it, it's going to be a fairly quick interaction, and it's going to leave. But if the T cell recognizes what's presenting, then you're going to use all these molecules to stabilize this interaction, and you can actually get that T cell working. So similar to dendritic cells, macrophages can engulf pathogens in similar ways. So it can either undergo phagocytosis or macropinocytosis. B cells are just a little bit different in that they recognize antigen right through their receptor, which is the membrane-attached antibody. And then once it's attached, it can engulf that way and then be processed and presented. So the B cell is one that's just a really a touch different. Okay, I think we've gone through this enough. But again, if the T cell receptor recognizes the dendritic cell and the MHC, the antigen and the MHC on the dendritic cell, then you can get a stable adhesion of those two cells together, so it can exchange signals. Adhesions made up of LFA1, which can bind to ICAM, but there's different types of ICAM, ICAM1, ICAM2, ICAM3. And then CD2, CD58 is another important interaction in this case. So the T cell receptor binds to MHC antigen. Remember, it has to recognize both the MHC and the antigen. And then the co-receptor, remember it's a co-receptor because it's going to also bind to the MHC, the same thing as the actual receptor does. And then you're going to have an adhesion to prolong the contact. So in order for this adhesion to be successful, you have to have 3 signals. And you need all 3. If you don't, then this interaction doesn't happen. So the first one is that the T cell receptor has to bind to recognize and bind to both MHC and the antigen. And then you have your co-receptor, which also binds to the MHC. And this is what we call signal 1. But this is not enough to activate a T cell, a naive T cell. You also need an interaction between CD8 and B7. So these are the co-stimulators. B7 has 2 types, B7-1, B7-2, also called CD80, CD86. I would know both of those terminologies. I would also know that it's CD28 on the T cell. I think of B7 on the B cell, which is right, the dendritic cell, but they're both antigen presenting cells. Maybe it's kind of a silly way to remember it, but that's signal 2. And then the necessary signal 3 is that the antigen presenting cell that's presenting is going to be sending cytokines to the T cell that's going to help it know how to differentiate. So you're going to have the T cell receptor interaction, co-stimulators, and cytokines all contributing to the fate of this T cell. Okay, so as the T cell starts to get activated, one thing that changed is its IL-2 receptor. So a naive T cell has a 2 subunit IL-2 receptor, gamma and beta. It has moderate affinity, but then once T cell's activated, you want that T cell to be replicating very quickly. And IL-2 is the essential cytokine for growth of T cells. So it actually ends up getting a 3rd chain on this IL-2 receptor, the alpha chain. So this 3-chain receptor then binds to IL-2 with high affinity, and it really causes that T cell to start proliferating. This is the same thing in just a slightly different picture. So the naive T cell has a 2-chain receptor. It can respond, but then once things get activated, it actually starts making a 3rd chain. And can actually respond in an autocrine manner in that it secretes both the IL-2 and displays the receptor, and then it causes it to these T cells to proliferate in a very robust way. Okay, so if the T cell let's see. So in this interaction, right, other things can occur because you don't necessarily want the immune system to run unchecked. So everything that gets the immune system kind of upregulated and going, there has to be something to downregulate it again. So there's actually a competitor to CD28 for B7, and that's called CTLA4, and this is the downregulator. So if an activated T cell is presenting to an antigen-presenting cell, in this case it's more likely to be a B cell or macrophage at the site of infection. It's still going to have the same interaction with the MHC and T cell receptor and co-receptor. But in this case, if that B7 doesn't get that co-stimulation and tells the T cells to stay activated, it can bind to the CTLA4, and the CTLA4 will actually start deactivating the T cells. This will actually start ramping down the immune system, presumably as things start to get cleared. The T cell is going to start upregulating CTLA4. CTLA4 binds with much more affinity B7 than CD28 does, and this is good. You don't want your immune system running unchecked. CTLA4 sitting next to CD28, the CTLA4 is the winner. This is the one that really binds with high affinity. So a couple other checkpoints for the T cell include the presence at all of that co-stimulator. So what can happen is that antigen can be presented, but if that antigen-presenting cell hasn't gotten any other signal of inflammation, then it is not going to have all its co-stimulators on the outside. So what is necessary is for the pathogen to be there, as well as there to be inflammation going on where that pathogen is, because that inflammation is going to send cytokine signals, such as a direct interaction with a toll-like receptor to that dendritic cell to upregulate those co-stimulators. So it's a system of checks and balances. If it's not there, so if it sees just a pathogen and there's no inflammation, well, to me that says what's more likely to be self-protein, right? There's not going to be anything else getting that going. What happens is that you have an interaction with the MHC and the T cell alone. You don't have all the co-stimulators, and that actually signals the T cell into either deletion or anergy. So then even if an anergic T cell sees an antigen again, it still won't go after it. You really need those co-stimulators there, and those co-stimulators are going to be there because of inflammation around the cells. Another fail-safe is that you absolutely need T cell receptor and MHC interaction. If the co-stimulators happen to bind, nothing's going to happen at all. It'll just press on. This is a necessary interaction that you really have to have that antigen being presented on MHC for that T cell to get stimulated. So T cells aren't just kind of stimulated, willy-nilly in a non-specific way. It's very specific how the T cells get stimulated and for how long. That all makes sense to everybody? Okay, so the whole process inside the lymph node, right, you have your antigen-presenting cell, in this case what's considered a dendritic cell, presents to the T cell, and it's going to give it 3 signals, MHC T cell receptor, co-receptor interaction, co-stimulators, and then there's going to be cytokines being secreted. And then you T cells are going to proliferate and differentiate based on signaling from IL-2 in an autocrine way. And after that, they're ready to leave the lymph node, go to the site of infection, and actually undergo their effector function. They're effector T cells at this point. So these are nice molecules that we can exploit to figure out if a T cell is naive or activated, and here's some markers you would see. Right, so if it's resting, it's going to have things like CD45RA or CYP1, which we talked about. Selectin, of course. These are things that are addressing it to the right place. Then once it's activated, well, here's some good ones like VLA4, the very late. CD485RO is going to get upregulated. We use these a lot when we're looking at immune deficiency. Activated should have RO. Always going to have a T-cell receptor if it's a T-cell. This is a nice little table to help you understand the changes on the cell surface of the T-cell that are occurring as it's switching from naive to effector cells. So if the T-cell that's activated is a CD8 positive T-cell, well, it has a very specific function. So it's a cytotoxic or killer T-cell, and it is going to be directly cytotoxic to an infected cell. So things that, it's usually viral infections that will activate a CD8 cell. Because once a cell is infected with the virus, you really need to kill that entire cell in order to clear the virus. But it does it in a way to not cause a lot of inflammation just from the cell death, like direct cell injury does by necrosis. It does it by something called apoptosis, or programmed cell death, so it can compartmentalize it and make it very neat. So nothing around the cell is disturbed as it's dying. It just dies in this package in a very clean way so as not to cause more inflammation around it. So once we start talking about how T-cells have differentiated, it's very tempting to think of them in a silo. And the whole immune system, I think, is one of the reasons it's so tricky to learn is that we're presented everything in these silos. This is what happens here, this is what happens here, this is what happens here. But you have to remember that this is all happening together. So all these things are helping each other. So even as CD8 cells go off and their function is to kill infected cells, they're still getting help from CD4 cells. So CD4 cells are probably helping stimulate the CD8 cells and keep them alive and functioning and going by different methods, one of which is cytokine secretion. But also sending signals through antigen-presenting cells to help those get going. So I kept this in here really just more as a point to never start thinking about this stuff as silos and doing something alone. Okay, so if it was a CD4 positive cell, then it could have several fates. So the ones that we know have known about the longest, Th1 cells and Th2 cells. Th1 cells, which we think fight intracellular bacteria, such as mycobacteria, TB. Then Th2 cells work through eosinophils and mast cells to kill helminthic parasites. Th17 cells work through neutrophils to kill extracellular bacteria. And then there's a specialized T helper cell called a follicular helper T cell that sticks around in the follicle of the lymph node. It doesn't leave. It never regains that CYP receptor. And its whole job is to stay and to activate B cells and help a B cell differentiate into what it needs to differentiate into. And then, of course, like everything, there's always the counterbalance. And in this case, it's T regulatory cells. So some cells turn into regulatory cells, which actually downregulate the immune system. There's two types. Ones that are generated directly from the thymus. But then there's also inducible T regulatory cells that in the periphery are induced to down-regulate infection or to not attack a certain potential pathogen. Again, probably in the case of self-protein. Probably it gets turned into a T regulatory cell if it doesn't see those necessary signs of inflammation after it's been presented a protein. Okay, So the differentiation is dependent on that third signal. Remember, signal 1 is T-cell receptor, 2 co-stimulators, and 3 are these cytokines that's being delivered by the dendritic cell. So the dendritic cell understands what type of immune response is needed to clear the infection because it's been at the site of infection. So remember, that's where it started, and it was getting other signals outside of just engulfing that pathogen. It was actually having direct interactions with its receptors on the outside, like toll-like receptors and all those other pattern recognition receptors that we've talked about. So then when it's presenting to this T cell, it's going to specialize what it's secreting to tell the T cell. So if the T cell, if the dendritic cell is secreting TGF-beta, so this is one to start getting plastered in your mind as a downregulator. So TGF-beta, it's a signal to turn this into a T regulatory cell. The T regulatory cell secretes more TGF-beta and IL-10, and these are the two downregulating cytokines that you really need to start recognizing as downregulators, TGF-beta and IL-10. So this TGF-beta, its job is actually to turn on these essential transcription factors that that seal the fate of the T cells to become a regulatory cell. The TGF-beta turns on the essential transcription factor FOXP3 in the case of T regulatory cells to make it a Treg. In the case of a Th1 cell, it's IL-12 and interferon. So interferon interferes with viral replication. So it's the virus Th1. So this is the viral pathway. This all should make pretty good sense at this point. Interferon IL-12 in itself is its own pathway. And it's going to turn on this essential transcription factor TBET and turn this into a Th1, and it's going to do its effector function through secretion of things like interferon primarily and IL-2, the growth factor. And then, of course, Th2 is going to turn into Th2 in response to IL-4. So presumably, if a dendritic cell has seen a helminthic parasite, it's going to start secreting IL-4 when it has this interaction. It's going to cause this essential transcription factor GATA3 to be expressed. It will commit this to a Th2 type cell, which will then secrete IL-4 and IL-5 as its effector function. Finally, Th17 cells are turned into Th17 by two signals, TGF-beta and IL-6. Essential transcription factor is ROR gamma T or ROR gamma tau. And then this Th17 cell works by secreting IL-17 and IL-6. I've actually skipped over one. Abbas never talked about these, so I know much less about these follicular helper T cells, but they become follicular helper T cells by signaling from IL-6 to this BCL6, this essential transcription factor. And then secrete things like IL-21 and ICOS. ICOS being inducible co-stimulator. So this is another stabilizing molecule similar to LFA1 ICAM interaction. You can get ICOS ligand, which makes sense because follicular excuse me, follicular helper T cells are going to be wanting to adhere to a B cell and get the B cell functioning and going. All right. So once the effector T cell is an effector T cell, right? It's going to exit the lymph node through the lymph. It's going to go through the thoracic duct, and it's eventually going to get back to the blood vessel. Once it's gotten to the blood vessel, it's going to get to the site of infection, and then remember it has to slow by a selectin interaction, and this is going to be EMP selectins. And then it's going to adhere to the vessel wall. In this case, it's going to be interaction with the integrin, VLA4, VCAM, very late. It's going to stop, and it's still going to exit via diathesis. And in this case, it's going to interact with an antigen-presenting cell at the site of infection. And in this interaction, a few things happen. So cells will actually flatten on one side, and they're actually rearranging their cytoskeleton in order to maximize the amount of surface contact that occurs. And there's actually a large part of the two cells that will be in contact with each other. And this entire area is called the SMAC. And then there's the P-SMAC and the C-SMAC. The P-SMAC is the peripheral part, and the C is the central part. So the central part is going to contain the T-cell receptor and the co-receptor and the co-stimulators. And then for the most part, this peripheral part is going to be all the stabilizing adhesion molecules. But there's going to be a whole bunch of interactions. So everything's going to have to come together in this one space where the two cells are touching in order for all those interactions to occur. So this is a drawing of the same thing, that here you have either an antigen-presenting cell, more likely a B cell or macrophage at the site of infection, right, or a target cell in the case of a CD8. So you're going to have this whole area together with the SMAC touching. For this to happen, everything has to rearrange and line up. And here's kind of your whole SMAC here. But everything's lined up. If you cannot line up, and this effector T cell can't make everything line up for all these interactions to occur, this is what happens in what's called Aldrich syndrome. So you have an immune deficiency that's less severe than SCID, but it's still a T cell problem. So in SCID, right, you have T cells, but since you don't have T cells, you can't get your B cells activated either. In this case, your T cells are fine while they're in the lymph node interacting with B cells. So your B cells get activated and do okay. So it's not a combined immune deficiency. It's T cell. It's when it becomes an effector cell and leaves the lymph node that you start running into problems. So you don't see the same severity as with SCID for the most part. So this is just one more table that pretty much says the same things that I've mentioned before, as in what the effector molecules of each of these T cells say. But just for a minute, we're going to talk about what happens if you have a CD8 positive T cell. And this is going to happen through the effector molecules perforin, granzyme, granulysin, and fast ligand. These are the things that are going to directly cause the cell death of another cell. Oops, forgot about the cell. This is actually maybe poorly placed. Sorry about that. But this is on page 371, 924. A very nice chart of all the different cytokines, and probably the vast majority of the cytokines you need to know for the boards are in this table here. I think this is a very nice summary. Okay, just skipping over that there, though it's there for you. Okay, so back to CD8 cells. So 3 molecules: perforin, granzyme, granulysin. Okay, so perforin, I think of as perforating a hole in the target cell. So it actually aids in delivering the contents of these granules into the next cell. And the contents include granzymes and granulysin. So granzyme is actually what's going to induce apoptosis once it's in the activated cell. And then there's another one called granulysin, which actually has antimicrobial properties, and in high enough amounts it can also induce apoptosis. Now I don't know if that's physiologically relevant or if that's just something that's been noticed in vitro, but perforin and granzyme, I would say, are the two that I definitely need to know. So perforin delivers granzyme actually starts the process of apoptosis. So there's a couple other things like cerglycin, which is really a scaffolding to hold these things together during delivery. But once it's delivered, the granzyme is going to start this caspase cascade, which we've talked about before. So apoptosis in this situation is mediated through a cascade of caspases. So just similar to how a complement is a cascade. It's a protein activates, protein activates, protein activates, protein. Same thing happens with the caspases. So caspase 3 is activated, and then this causes a chain reaction, which results in the release of cytochrome C from the mitochondria into the cytosol. So once this happens, then you get fragmentation of your DNA, and you get blebbing of all the rest of the cell, and it's packaged very nicely to be processed by other phagocytes in its environment in a nice, neat, tidy, inflammation-free way. The other option, and I'm pretty sure there actually isn't a not a great slide for this, is that there's a direct interaction between FAS and FAS ligand, CD95 ligand, which is just another pathway to start apoptosis. If there is a defect in either CD40, CD40 ligand, then you may have an immune deficiency called ALPS, autoimmune lymphoproliferative syndrome, where you're going to have hepatosplenomegaly. You're not going to be able to get rid of cells that need to be get rid of, so you're just going to house them. Right, so just very quickly, this makes the important point that even in cells in very close proximity are not going to be injured by this process. It's very neat, it's tidy. There's no inflammation caused by it, so it can die off by apoptosis with the area around it not being affected at all. Okay, so back to CD4 cells.
If you are,
I am. I think, therefore I am.
So Th1 cells, so remember, these are the cells that are going to take care of intracellular bacteria. So intracellular bacteria being mycoplasma, for example, like tuberculosis. So in this case, the T cell is going to interact directly with the macrophage. The macrophage is going to act as an antigen-presenting cell. So in the environment, this is an effector cell. So it's not a dendritic cell anymore. It's a macrophage. And you're going to have very similar interactions, but one of the important interactions in this case is CD40 ligand CD40. And the T helper cell's main job, remember, is to secrete cytokines. So interferon interferes with viral replication, and it really upregulates macrophages, and it helps macrophages to destroy what's sitting stagnant inside of it. So an activated macrophage upregulates its whole killing process. So remember we talked about the phagocyte oxidase where it's undergoing an oxidative burst. So this whole process gets upregulated and makes this macrophage more efficient at eating and killing what it needs to kill off. Yeah, I don't think anything I haven't said is here. So that's about it. So, right, if this was a T helper cell, it's a Th2 cell or Th17 cell. You know, the process is going to be very similar in that it's an effector cell, it's at the environment, and it's secreting cytokines that are going to direct the relevant other cells, you know, to do something, to do what is necessary to clear that infection. In a very similar way. But that's it, unless there's any questions. Next time we're going to talk, Chapter 10 is the humoral response, so it's going to be the same thing, but talking about how B cells get going, or start as naive B cells, get activated, and then undergo their response.